I can't hear you. Post the exam answers, or? The previous exams are posted on Blackboard, right? That's part of the study guide. Right? Everyone gotten hold of the study guide? Very important. Look at those previous exams. Use those exams to test your knowledge. This exam will be quite similar to previous exams. Okay? So it's posted on Blackboard as the study guide, and it has the last several years of exams with the answer keys. Okay, any other questions? Okay. So last time, what I'd like to do today is finish talking about signaling and growth control, this new type of signaling pathway, the elucidation of this signaling pathway in large part owes cancer biology, cancer genetics studies, and biochemistry, cell biology, to come up with novel modes for attacking a signaling pathway. And last time I talked about the identification of the first oncogene, the first transforming gene that was found in what? Anyone remember where this gene came from? Yeah. Yes, good. There's a virus that infected chickens, Rous sarcoma virus. Harold Varmus, Mike Bishop, isolated the gene, demonstrated that that gene was both necessary and sufficient for transforming fibroblasts into cancerous cells, uncontrolled mitogenesis. And that gene, Mike Bishop hypothesized, was a mutated form of a cellular gene that through evolution the virus picked up and mutated, and when reintroduced into cells in a dominant way caused cell transformation. And he showed that there was a normal cellular gene, which he called, so the viral oncogene was called VSARC, viral SARC gene, and the normal gene was called CSARC for cellular SARC, the normal cellular gene that is not mutated, not transforming, and presumably plays some normal role in the cell growth control pathway. Okay. What you see here is a three-dimensional structure of the normal cellular SARC gene. The viral SARC gene is an oncogene. The cellular SARC gene we call a proto-oncogene. Okay. It is not oncogenic, but it gives rise, in the case of a virus, it gives rise to the viral gene, which is an oncogene, and you'll see other ways by which this gene might become oncogenic. Okay. So it has several interesting, its overall folding has several interesting components that give rise to the folding of this protein, and we're going to talk about that in this lecture. It contains the catalytic domain, which we'll discuss in a few moments what that catalysis is. It has an SH2 domain, which stands for SARC homology domain, which plays an important role in many proteins that are involved in signaling processes, and we'll see how. An SH3 domain, as well as an activation segment. So all of these terms indicate some kind of functionality, right? And it's important that we understand it's one thing to isolate the gene and isolate the protein and even crystallize the protein and understand the structural configuration of the protein, but it's a whole nother thing. What we really want is the functional significance of this protein. What is the functionality of this protein? What is it about the viral SARC gene that when introduced into cells causes uncontrolled growth? And what kind of functionality does cellular SARC have in normal cells in growth control? The first clue to the functionality, really the first demonstration of a functionality for the viral Sark protein, came from Ray Erickson's lab, in which he purified the protein that was synthesized off of the viral Sark gene, off the virus. He purified the protein, and he put it in a test tube, and he started looking for some kind of activities. And the one activity that he identified came about when he put ATP in the test tube together with purified Sark and a mixture of cellular proteins. And he found that when the ATP was made radioactive, it transferred radioactivity to a bunch of cellular proteins. Okay? So in the presence of ATP, cellular proteins were able to become radioactively labeled in the presence of viral Sark. What does that suggest? Yeah. Yes, excellent. It's a protein kinase that phosphorylates proteins. Okay. That was a major discovery and got him into as a member of the National Academy of Sciences. He's on the list of people that are candidates for a Nobel Prize. It was extremely important because it pointed to the first oncogene as being part of a regulatory pathway. Everything that was known about protein kinases had come really from the work of Krebs and Fischer with cyclic AMP-dependent kinases and the kinases involved in glycogenolysis. And now growth control, a key component of the growth control process, which caused uncontrolled growth, was a protein kinase. Okay, suggesting that protein kinases played a wide role in mediating changes in cellular physiology in a wide range of processes. And the question was, what is the substrate for the Sark kinase? Okay, so it's a protein kinase. We take the viral Sark, we put it into cells, and what is it phosphorylating? And the first indication that this was a unique type, a new class of protein kinase, came from the work of Bart Sefton and Tony Hunter working at the Salk Institute in San Diego. And what they found, they developed a method for separating phosphoserine and phosphothreonine and phosphotyrosine from one another on thin layer plates. Okay. And why would they look at those three amino acids? Yeah? Excellent. Okay. Each of the amino acids, these are the only three amino acids that have a hydroxyl group on their side chain and which are phosphorylatable by a protein kinase. And so they just simply wanted to determine which amino acids it had more specificity for, thinking that it was phosphoserine or phosphothreonine, just like the other protein kinases that had been identified to date. And what they found was extremely surprising. And this experiment shows control cells in which which have been fed radioactive phosphate so that all the ATP becomes radioactive, and kinases could transfer their phosphate to proteins. Okay, and then we could detect those proteins that are phosphorylated, and if we cleave those proteins into the individual amino acids and run it on a thin layer chromatogram, we could separate those phosphorylated amino acids from one another, and the ones that show up dark were exposed to an X-ray film because they're radioactive. So the radioactive spots indicate where P32, where radioactive phosphorus is located. And in normal control cells, you see that phosphoserine is the predominant, followed by phosphothreonine. So in normal cells, there's a lot of phosphoserine and phosphothreonine, and there's little, if any, detectable phosphotyrosine. Okay. However, if you now look at VSARC transformed cells in which we've introduced the viral SARC gene, say we infect cells with Ralph sarcoma virus and allow a lot of production of the viral SARC gene, and then feed those cells radioactive phosphate, and now look at the amino acids that are phosphorylated in those cells, one sees that phosphotyrosine essentially takes over as the predominant amino acid that is phosphorylated. What does that suggest? Yes? A mutation? What kind of mutation? A mutation of phosphoserine and threonine into phosphotyrosine? Okay. Anyone else? Yeah? An exchange of amino acids caused by what? Yes. Which gene? Viral SARC. So all we're doing here is taking cells that now have expressed viral SARC and looking at the amino acids that are phosphorylated. And what we see is a switch from the normal serine and threonine to predominantly tyrosine. So tyrosine now becomes the predominant amino acid. Why? Yeah. Okay, so we're getting very close. So he's saying something about viral SARC is causing tyrosine to become a major amino acid phosphorylated in these cells. Yeah. Yes, beautiful. The viral SARC is a protein tyrosine kinase, meaning the specificity for SARC is tyrosine. Specificity for cyclic AMP-dependent kinase is serine and threonine. But the specificity for this new kinase, this growth control kinase that causes cell transformation, is tyrosine. And when we infect cells with the viral SARC, express the viral SARC gene, we get a hell of a lot of kinase activity that's directed at tyrosine. That doesn't mean, so maybe what's misleading about this is it doesn't mean that we've lost serine and threonine. It's just that at similar exposures, serine and threonine become minor relative to tyrosine. They're still there in the same amounts as before, but to get an exposure which looks reasonable, it's almost all phosphotyrosine relative to serine and threonine. So we've introduced a protein tyrosine kinase into cells that is responsible for cell transformation. This was a major finding, simple experiment, but a major finding for its implications, which are that there's a new class of protein kinase that recognizes tyrosines instead of serines and threonines, and that's responsible for uncontrolled growth as an oncogene. Okay? So clearly, we'd like to understand now what is the mechanism by which the normal cellular SARC gene folds. Okay. What is the kinase activity of the normal cellular SARC gene? And what is it about the viral SARC gene that differs from cellular SARC through mutagenesis that causes it to have such a dramatic effect in cells? So I should mention that if you repeat this experiment, introducing the cellular SARC gene, essentially overexpressing more of the cellular SARC gene in cells. Cells normally have cellular SARC. If you put more of it in, you don't see any significant change in the phosphoamino acids that are produced. What does that suggest about cellular SARC? We put normal cellular Sark into cells. We don't see this type of change of a dramatic increase in phosphotyrosine in cells. And cells, of course, have normal cellular Sark. Yes? Okay, it needs more to activate it. What does that mean? Forget about that it needs more, but if it needs something to activate it, means that it's not very active. Okay? Good. So it's not very active. It is not an active tyrosine kinase. So if we put cellular Sark into cells, we don't see much of a difference. It is not normally very active. So the difference between cellular Sark and viral Sark is the level of activity. One is an extremely active tyrosine protein kinase, and the other is inactive, relatively inactive, in the normal cellular form. Okay, so now let's look at normal cellular Sark, which I showed you the crystal structure of, with all the domains and how they fold on one another. And here's a cartoon of that. And we see that there are several domains that are very important to protein folding and functional activity. The first domain of interest is the C-terminal domain, the carboxy-terminal domain, which contains a tyrosine 527, which is normally phosphorylated in cells. So under unstimulated conditions, normal conditions with normal cell growth, this tyrosine is constitutively phosphorylated. It is always phosphorylated. And it has a high interaction with a domain called the Sark homology 2 domain. The SH2 domain, which is found on a number of proteins, first discovered in Sark, binds tightly to phosphotyrosine with the proper surrounding sequence. So in normal cells, SH2 domain is bound to the phosphotyrosine at the carboxy terminus. Another important folding occurs with what's called the SH3 domain, the Sark homology 3 domain, with an internal region that's proline rich. So we have folding, tight folding of the protein, such that the SH2 domain binds the phosphotyrosine on the C-terminal, and the SH3 domain binds to a proline-rich domain, and the rest of the protein is involved in the catalytic activity of the protein kinase. Okay, and the very active site contains another tyrosine that is well folded within this tightly folded, overall folded structure. In this normal configuration, this normal folding of this protein, the kinase activity of the catalytic domain is inactive. So if we take this protein and we introduce it into cells, you have an inactive kinase that remains inactive in normal cells, and it won't have any change in the level of phosphotyrosine. If we unfold this protein, the kinase activity will become activated. Okay. So let's look now at the structure of the viral Sark protein to try to understand what it is about the viral Sark protein that's different from the cellular Sark protein that causes it to be extremely active as a protein kinase. And that's seen very easily when we compare the structure, the sequence of cellular Sark to that of viral Sark. And what we see is that the viral Sark gene, a prominent difference between viral Sark and cellular Sark is that the viral Sark gene is missing this carboxy terminal domain entirely. What effect would that have? And why? Yeah. It wouldn't be phosphorylated, yes. So it's missing the tyrosine and it won't be phosphorylated. And so what's the consequence of that? That's good. Yeah? It wouldn't be inactive, meaning it would be active. Excellent. Okay. So if it's missing this phosphotyrosine domain, the SH2 cannot fold properly by binding to what's missing, right? If it can't bind the phosphotyrosine because it's missing, you can't have this folding. And as I mentioned, the unfolded protein becomes active, okay? Because the folded structure is inhibitory to the catalytic activity, and the unfolded structure frees the catalytic domain to be fully active. Okay. So the predominant mechanism or mutation which has caused viral Sark to become hyperactive or fully constitutively active, continuously active as a protein kinase is the loss of the ability for this folding to occur. Okay? So what does that suggest about how C-Sark might normally be activated? Yeah. Sorry? Okay, excellent. So one possibility is dephosphorylation. So we'd like to know what is the mechanism by which this tyrosine is kept phosphorylated. And it turns out there's another protein kinase called CSK or C-Sark kinase that phosphorylates constitutively in normal cells, constitutively phosphorylates this tyrosine. And if we remove CSK from cells, that tyrosine becomes unphosphorylated and Sark becomes activated. So one way of activating this kinase is by dephosphorylating this or preventing its phosphorylation. Okay? What's another way that we might activate this protein? Yeah. Sorry? Okay, well that's what, cleaving the C-terminus is what has happened. So clearly that's a way. What's another way? Yes? Okay, changing the tyrosine, so a mutation that might change the tyrosine to another amino acid would do it. That's good. What else? Stop thinking about the C-terminus. What else? Yeah. Sorry?
Extreme conditions like extreme heat will unfold the protein.
Okay, he's saying extreme conditions that would unfold the protein. We don't need extreme conditions. We can unfold the protein in very specific ways. That's good. How? Yeah. Sorry? Okay, that's an extreme condition, would be removal of the SH2 domain. Good. Yeah? Okay, good. So mutations, additional mutations that might interfere with this folding process, or really anything that would interfere with the normal folding. If we disrupt the SH2 interaction with the phosphotyrosine, if we disrupt SH3 from interacting with the kinase domain, if we disrupt the folding, we cause an unfolding of the protein, and anything that would cause an unfolding of the protein would activate it. Okay, so C-Sark can be activated by a variety of possible measures that would cause a disruption of the normal folding. We could disrupt the SH2 domain interaction, disrupt the SH3 domain interaction, mess around with that phosphotyrosine. All of those will cause an activation. Okay? And what we've seen is that the major activation mechanism for VSARC was the removal, the truncation of that whole terminal region containing the phosphatyrosine. So now we understand we've uncovered a whole new class of protein kinase that plays a critical role in cell growth control. We can mutate this protein and cause it to become an activated protein kinase, which now acts in a dominant way to cause uncontrolled growth. Now, what do I mean by a dominant way? Well, if I have Rous sarcoma virus, which has the VSARC gene, or I just isolate the VSARC gene and introduce it into cells, those cells have a normal cellular SARC gene. But by introducing this activated form of a mutated gene into cells, it acts in a dominant way over normal growth control to cause uncontrolled growth. So it's a dominantly acting mutant oncogene that acts in a dominant manner on the growth control process and is dominant over the proto-oncogene CSARC. Okay? I know that mutations, a variety of mutations that I could introduce into CSARC would cause it to become oncogenic if I could cause uncontrolled growth through unfolding. One of those type of mutations is what VSARC has done, but any of the disruptions of the folding would also achieve similar effects. Now that leads to a very important and interesting hypothesis, and that hypothesis is, or the question is, that cancer may be caused, in general now, cancer may be caused by a mutation of a cellular gene that causes it to become dominant over the normal growth control process, such that you have abnormal growth control. Does everyone get that? So the question is, by example of what has happened with Rous sarcoma virus and cellular SARC and VSARC, one may make the hypothesis that cancerous growth could be caused by mutation of a normal cellular gene, causing it to become uncontrolled and dominant over normal cellular growth control process, such that you get uncontrolled growth. So growth is normally governed by a growth control pathway, and if I take a component of that growth control pathway and I disrupt it and make it, in the case of SARC, hyperactive as a kinase, I will now cause uncontrolled growth. That was the question. So we're going now from the example in a virus that causes tumors in chickens, Rous sarcoma virus, and identifying the genetic basis of that and the biochemical basis of that, and seeing whether that reflects a general understanding of growth control processes in human cells underlying cancer. Yes?
You said that it controls the growth. So if it controls the growth, then their cells will be replicating more through mass action in comparison to other cells. So it would just be more replication of DNA. So once it's replicating the DNA, that protein will also be replicated. So then if you just keep on doing sequential steps, then eventually that won't be the most dominant piece of culture.
So we don't need to think about populations of cells in order to understand the dominance. It's really at a molecular basis in a single cell that we're talking about dominance. Okay, yeah.
Is it like more stable? Is that mutant more stable than the regular configuration?
Is the mutant more stable? The mutant is active, and the normal cellular gene is not active. And by having this constitutive activity, we constitutively cause uncontrolled growth. Okay, good question. Okay, so how do we answer this question? Well, the question, would be answered by seeing if in a normal oncogenic or normal cancerous human cell, there is a gene that is expressed in that cancerous cell that causes uncontrolled growth in a normal cell. Okay? You have a question? Sorry? Proto-oncogenes? I'm not sure what you mean. It's the oncogene that's dominant. The proto-oncogene, when it becomes oncogenic, becomes dominant. That's the idea. Okay? And that question was addressed by Robert Weinberg. It was actually addressed simultaneously or in parallel by several groups, and the first to get the answer was Robert Weinberg's group at MIT. And what he did was the following. He took a human tumor cell line. He took cells from human tumor and isolated the DNA from the human tumor. And he asked the question, if I transfect this DNA into normal mouse fibroblasts, can that expression of that DNA cause uncontrolled growth of those fibroblasts such that they become cancerous? Okay? And that was allowed at the time that there was methodology for introducing DNA into cells. So technological advance, the ability to introduce DNA into cells to get it expressed in other cells, allowed for this question to be answered. And so what he did was he took human tumor DNA, which presumably, hypothetically, contains an oncogene in it, which is why it's a tumor, coming from a tumor. And he introduced the tumor DNA into normal fibroblasts, which grow contact inhibited, attached to the dish, anchorage dependent. And he looked for cells expressing the human DNA that would now grow as cancerous cells uncontrollably in 3 dimensions and not anchorage dependent. And so he found, in fact, that there were colonies of growth that mouse fibroblasts that had taken up the human tumor DNA and expressed it were now growing uncontrollably, demonstrating that human tumor DNA contains an oncogene in it that acts in a dominant manner and can act even in mouse cells in a dominant manner to cause uncontrolled growth, just as we've seen Rous sarcoma viral infection of normal cells causes uncontrolled growth by introduction of an oncogene. Okay? And so the next step would be what? What would you want to do next? Yeah. Yeah, beautiful. Try and identify what that oncogene is. Now, how do we identify it? Well, we have a method by which we could take DNA or fragments of DNA, introduce it into cells, and we have an assay for looking at its oncogenic activity. And so smaller and smaller fragments of the DNA would be introduced into cells and identified on the basis of its causing uncontrolled growth until we identify and clone the gene that is the oncogene in this normal human tumor. Yeah. So, good question. So he's a technical question, and that is, what is the size of the DNA that was used in the initial experiments? Well, you can't take entire genome and transfect it in. So what you do is chop up the DNA with restriction enzymes and introduce all of the fragments of DNA into the total population. And the ones that lost contact inhibition are the ones that you proceed with. And so you go through multiple rounds. You now take this colony, isolate the human DNA in those mouse cells, and make smaller fragments, redo this again, and then you end up with a single or a bigger chunk of DNA with multiple genes, clone it out, and then find a clone that will do the same thing, and you have your cloned gene. Okay. So he cloned the first human oncogene, which puts him, not only makes him a member of the National Academy of Science, puts him on the list. He is receiving multiple awards, and he's on the list for a Nobel Prize. He is a Nobel Prize candidate. Okay, so in the coming years, we might see him win a Nobel Prize. The first human oncogene. So just as we approached the viral Src gene in Rous sarcoma virus, we're going to approach this gene that came out of human tumor DNA. What is this gene encoding? What is the functionality of that protein that this first human oncogene encodes? And this is the protein. It's called RAS for reasons I won't get into. The RAS protein, the RAS gene, was the first human oncogene. Okay. There is a normal cellular counterpart to the gene, and then there is an oncogenic form, mutant form of the gene. Right? The idea was that human tumors contain an oncogene that is a mutated form of a proto-oncogene that now causes uncontrolled growth. The normal RAS protein is a protein that's nicely folded. We have 3D structure of this protein. And in this particular configuration, we see that this protein is bound to GTP. Isn't that interesting? A GTP binding protein. Okay. A GTP binding protein is reminiscent, okay, of a G protein, a GTP, a guanyl nucleotide binding protein. And the question is it similar to the G proteins that we've talked about until now? And if one compares the 3D structure of a G protein, in this case it's transducin, G sub T, to RAS, we see that there is a portion of transducin, the GTP binding portion of transducin, of the G alpha subunit, that looks very, very much like RAS in its overall configuration. It is, in fact, what we call now a small G protein. It is a G protein that encodes the functional domain of the alpha subunit of G proteins. Okay? Of course, it's a different gene, it's a different protein, and it is the first member of a class of small G proteins. There are many genes that encode small G proteins that play functional roles in a variety of processes. Okay, so the first human oncogene is a G protein. What does that mean? If it's behaving like the G proteins that we've been talking about thus far, in its inactive conformation, it would be bound to GDP. Upon guanyl nucleotide exchange, we would exchange GDP for GTP, and we would end up with an active form of the G protein. That active form of the G protein would now engage signaling pathways. Okay? And it would have an intrinsic GTPase activity, causing GTP hydrolysis and inactivation. These processes, the guanyl nucleotide exchange and the GTP hydrolysis would be, would be mediated by a guanyl nucleotide exchange factor and a GTPase activating protein. That's the normal G protein cycle that we've seen. And is it the normal cycle that we see with the small G proteins? This is again another illustration of that cycle. We have guanyl nucleotide exchange factors mediating guanyl nucleotide exchange, and GTPase activating proteins enhancing the GTPase activity. So it's a reversible activation, deactivation process. Okay. And RAS is a small G protein. And yes, this is part of the normal RAS activation cycle. This is the normal RAS activation cycle. It is activated and deactivated in much the same way as we've seen for other G proteins. So the first human oncogene turned out to be a new class of G protein, a small G protein. Now, I'd like to just talk about a couple of very interesting mutations that were found in this G protein. One is an activating mutation. Okay. And this is similar to what we've seen with the toxins' effects on G proteins. An activating toxin, like cholera toxin, would affect this cycle, and a mutation, an activating mutation, would similarly affect this cycle. And that active mutation would be a mutation which blocks the intrinsic GTPase activity. Okay, so if you block the GTPase activity, you remain in the active configuration, GTP bound, and you're constitutively active. So one mutation, an activating mutation, would be a mutation that blocks the GTPase activity. An inactivating mutation would be one in which the nucleotide exchange, the ability of the protein to bind GTP, is blocked by mutation. So let me ask you a clicker question, and that is, which of these two types of mutations, which of these two mutations do you think was contained in the first human oncogene? So the first human oncogene was a mutant form of RAS, and I've just shown you an inhibitory and an activating mutation. And the question is, A, is it an inhibitory mutation? Or B, is it an activating mutation that has caused RAS to become an oncogene? Does everyone understand the question? A is inhibitory, B is activating. Okay. Would anyone like to defend one choice or the other and explain why a particular mutation would cause it to become oncogenic? Yes.
I had it be the activating mutation because it stops the G protein from being activated, and it will continue to grow, and it will continue to eat the GTP.
Okay, good. So what he said is he believes it's the activating mutation because now you have a constitutively active RAS, and if it's involved in growth control pathway by analogy to SARC, the active form would cause uncontrolled growth. Anyone else? Yeah.
I chose yes while it's activating because once you stop the GDP process, so it will always stay active, and then as the effector, well, it also depends on like the function of the effector actually. If the effector
Yes, excellent.
affects the growth, then that will make it proliferate.
Okay, so what you're saying, what she's saying is, give your name. What she's saying is that it really depends on what the effector is, okay, and how that effector plays a role in normal growth control. Okay, so if that effector is a positive effector which mediates division, then uncontrolled activation of that effector would stimulate division uncontrollably. Anyone else? Anyone who picked the inhibitory? Change your mind? Yeah. Okay, very good. Very nice. So what she's saying again is it depends on what the role of the effector is. Okay? If you have, if you inhibit that ability of the effector to be functional and that effector is stimulating growth, okay, then you're going to get more uncontrolled growth. The key here, though, is understanding that it acts in a dominant way. Okay? So while you might envision either an activating or an inhibiting mutation to cause uncontrolled growth, only the activating mutation would allow you to act in a dominant way. So remember, if I introduce the mutant form of RAS, Weinberg showed, if I introduce the mutant form of RAS into normal cells containing the normal cellular form of RAS, it acts in a dominant way to cause uncontrolled growth. If that mutation blocked RAS function, then introduction of that into normal cells would be very difficult to block in a dominant way to overcome the normal growth control by the wild-type protein. Okay? Now, it turns out that an inhibitory mutant, this type of inhibitory mutant of RAS, could in fact act in a dominant way under special circumstances. Can anyone think of that? Of what circumstance? What we have is a mutation that blocks the ability of this protein to bind GTP, and I introduce it into cells. How could it block the endogenous normal cellular function of RAS? How could it be dominant? That's a tough one. Okay. Yeah. Sorry? No, we're not adding any more toxins. All we're doing is introducing this mutant into cells, and I'm asking, how could it be acting in a dominant inhibitory way? Yeah.
Is it possible that RAS could be acting as a repressor? So if it's inactivated, then you won't have checkpoints, and the cell will just keep on growing since this is not active?
Well, the thing is, the normal cellular gene is there and active. Okay, so we're introducing a dead form of RAS, an inactive form of RAS. How could it act in a dominant way to inhibit normal cellular RAS? Okay, I'm going to tell you. It's, yeah, one last try. So it might affect folding of other proteins. That's getting into a more complicated scenario. One last.
Well, if you produce an active form of RAS, then it wouldn't be able to interact with the effector molecules, and that could
Okay, you're very close. Very nice thinking. He said, well, maybe because it can interact with effector and it has something to do with interaction with effector. In fact, it's the other side. It can still interact with the guanonucleotide exchange factor. It's just that that interaction, because of the mutation, it's unable to nucleotide exchange. But the interaction is still possible. And if you have a dead form of RAS, a non-activatable form of RAS, interacting with GEF, if you have enough, if you overexpress, the more and more you express of the inactive form, the more and more of GEF you tie up and prevent it from interacting with the normal RAS. So if I express 10 times the amount of mutant RAS over the normal level of RAS, then 10 times the amount of GEF is now interacted with in a non-functional way, and you tie up all the GEF so that it can't activate normal RAS again. So in that way, it could act in a dominant inhibitory way, and that's been one really important tool for understanding the role of what types of effectors there are for normal RAS. Okay, so now the question is, we have two oncogenes that we've identified. We've identified SRC, for which we know there's a normal cellular SRC that can be mutated to become oncogenic. We know that there's a normal cellular RAS that can be mutated to become oncogenic. And the question is, are they part of the same growth control pathway, or are there multiple growth control pathways that each one plays a distinct role in? Are there parallel acting growth control pathways, or are they part of a single common pathway? And in order to answer that, the following type of experiment was done. We have two tools in hand. We have an activating mutant form of each of these viral or oncogenic forms of the protein. So we have an oncogenic SRC, which is hyperactive or constitutively active. We have an oncogenic RAS, which is constitutively active. Okay. And we can put each one into a cell, and it can cause uncontrolled growth. So we have a nice assay for their activity, physiological assay for their activity. If we introduce an active form of one and block the activity of the other, we can now ascertain whether they are components of the same pathway, and we could also determine the ordering of those components. So for example, in the example shown here, we introduce active SRC and at the same time inhibit RAS. In this case, in the early experiments, it was done by injecting an antibody of RAS into cells to knock out its function. Another way of doing it would be the dominant inhibitory form of RAS. Okay. So we activate SRC, we block RAS, and ask whether mitogenesis is caused by SRC is blocked by blocking RAS. Or we use an activated form of RAS and block SRC and ask whether mitogenesis through RAS is blocked when SRC is inactivated. And the results are that SRC's ability to induce mitogenesis was blocked when you blocked RAS, but RAS's ability to induce mitogenesis was unaffected by blocking SRC. So what does that mean about the relationship between these two components? Yeah. Yes, excellent. So RAS is downstream of SRC in a common signaling pathway. Okay? Because only if RAS is downstream would blocking RAS be able to block SRC action, but blocking SRC would not be able to block RAS action because RAS is downstream in the pathway. So this type of analysis is used today as well, activating, inhibiting mutations or biochemical analyses where you activate at one site, inhibit at another site, and determine whether components are part of a common pathway and the ordering of those components. Okay. So we know that this tyrosine kinase, SRC, as a tyrosine kinase is upstream of RAS. Somehow this tyrosine kinase is influencing GEF to turn RAS on, and we need to understand that. And we also need to understand what the active RAS is doing. So we have a tyrosine kinase influencing RAS function in a way that still needs to be defined. And we have RAS function somehow causing, downstream, somehow causing uncontrolled growth. And so the question is, what are the RAS effectors, right? The same way we needed to understand what the G protein effectors are. The G protein effector for adrenaline is adenyl cyclase, okay, and works through second messengers and amplification. What is the downstream effector of RAS in this growth control pathway? And the answer to that came from the similar type of approach that I've described, okay, for how RAS and SRC were initially identified. Okay. What would that approach be? How would you identify other components of this pathway? Yes? Sorry? Radioactive GTP. So you're now probing RAS itself. Okay? GTP doesn't get transferred to the effector. So how would I identify other components of the pathway? There are actually 3 components that I'll describe in a minute. But how are these components identified? How would you go about identifying them? Yeah.
By inhibiting RAS, and then doing, like, using them, like, from the downstream and upstream, inhibiting RAS, and taking one of the other factors.
Well, the question is, what other effector? I don't have any effectors in my hand. So you're sort of describing something that I can do later, but how do I get an effector? How can I identify additional effectors in this pathway? Yeah.
Knock out a gene and then put something else to replace it to see what causes it, like what's in the middle.
Knock out what gene? That's the question. Yeah.
SRC is a tyrosine kinase, so where would you want to find out what it phosphorylates? What's the next
Okay, so you want to know what's in between these, but what I'm asking is what's downstream of RAS. Okay? You're going to kick yourself if I tell you the answer. How did I discover SRC? How did I discover RAS? How will I discover additional members of this same growth control pathway? Yeah.
Do like one cell with the RAS activated high level and one without, and then do like a protein assay to see if there's like new band protein or more protein.
Okay, so that's an interesting approach. It starts to get at sort of what is the biochemical, what could RAS be biochemically interacting with. That's very good, and we'll get to that in a minute. One last attempt. Yeah.
You can also filter out the RAS by taking one cell that has the RAS receptor and clearing it out and then
Okay. So everybody is attacking this by using SRC or RAS as a tool. But that's not how these oncogenes were first defined. The way these oncogenes were first defined is there was some tumor DNA, there was some mutation in a normal cellular gene that when mutated caused uncontrolled growth. That gene was isolated and defined an oncogene. So RAS came out of a mutation that caused uncontrolled growth. That mutation was found to be in RAS. So let's look at other tumors. Let's look at other viruses. Let's look at other tumors for cancer-causing oncogenes, okay? And see if we always come up with SRC and RAS, or will we come up with the downstream components? And the answer is you come up with other components of the pathway. So the more tumor DNA you analyze and identify the cancer-causing gene, the more you identify additional genes in a common growth control pathway, right? By using cancer genetics, you can identify genes that are part of a normal growth control pathway that, when mutated to become hyperactive, cause uncontrolled growth. Question? So she's suggesting that we look at what genes, so you're assuming that genes that are involved in this growth control pathway are all part of the same operon, or all controlled along the same piece of DNA. That would take another 20 years of advanced technology over where we were. And the answer to that is no, they're not. They're on different chromosomes, different locations. Okay, but it's good thinking. Okay. So what I have here listed are 3 additional components of this growth control pathway that were identified on the basis of them being oncogenes when mutated and activated. Okay? And they contain a normal cellular inactive counterpart, which then becomes mutated. And we determine their ordering with respect to one another in the same way that I just showed you we determine the ordering between SRC and RAS. If I introduce an activated form of RAS and an inactive form of one of these downstream effectors, then I will determine whether or not these effectors are downstream from RAS or not. If they're upstream, blocking them wouldn't affect RAS-mediated proliferation. If they're downstream, each one of the blocks of these effectors will block RAS function. If I use an activated form of this effector and block a downstream effector, I similarly order them with respect to one another. And these 3 genes are called RAF, MEK, and ERK, or MAP kinase. They are all protein kinases. So, yeah?
So if you block RAF and you see that MEK works, it still produces like uncontrolled growth, like wouldn't that, like how will you know that RAS is needed then for the pathway to go downstream? Because if you can just use MEK to get the uncontrolled growth, like what is the purpose of RAS?
So if you could use an oncogenic MEK to cause uncontrolled growth, what is the purpose of RAS? The purpose of RAS would then become, in cells in which I don't have a mutant of MEK, in normal growth control, the only way MEK will be activated is through this whole pathway involving RAS. Okay? So the normal growth control pathway, where no mutations exist, lies in a tyrosine kinase upstream of RAS, and downstream of RAS, a kinase cascade, a cascade of 3 kinases, one activating on the next to activate the next by phosphorylation. And this is what a growth control pathway uncovered using cancer genetics, a combination of cancer genetics and biochemical interactions, looking at the biochemical interactions between each of these members, uncovered this pathway and the specificity of how this pathway acts. And of course, now the question is, certainly we'd like to know what the downstream effectors of that kinase are. We'll talk about that. But now we'd like to direct our attention to get a complete picture of this control pathway, of the players in this pathway. We'd like to know how tyrosine kinase affects RAS activation. What is at the top of this pathway? Okay, is a tyrosine kinase, SRC or other tyrosine kinase, at the top of this pathway? What mediates normal growth control? What is the signal that initiates this pathway? And the answer to that came not from studies of cancer, but came from studies of normal growth control. And that was through the identification of the first growth factor. So people working in how cells normally grow, like how fibroblasts divide and then stop dividing, reach their morphological changes, were growing cells in culture and then adding blood serum in order to stimulate growth, because there were factors, in quotes, in blood serum which could stimulate cells to divide. And one of those people studying this was Stan Cohen at Vanderbilt University. And Stan Cohen identified the first cellular growth factor, which he called epidermal growth factor, because initially it was found to stimulate epidermal cells to divide. And he purified, sequenced, determined the structure of this growth factor. And this growth factor, again, is acting from outside the cell, and we're binding to receptors on the cell surface to stimulate growth. And the question arose whether these growth factors, the first of which was EGF, epidermal growth factor, but there were a number of others that were identified for different cell types. The question was whether or not these growth factors were using the RAS effector pathway to mediate, to stimulate mitogenesis. Were they part of the same pathway? And to answer that is the same way we've answered all the other ordering issues, and that is we know that if we add the growth factor to cells, they'll divide. If we add the growth factor to cells and block RAS function, will that block the growth factor-induced cell division? And the answer is yes. So the first link between the people doing growth control and cancer genetics came from the demonstration that downstream of growth factor action lied this growth control pathway uncovered by oncogenes and proto-oncogenes. What that would mean if one is a cancer geneticist is that, well, if that's true and the EGF receptor is upstream of RAS and RAF and MEK and MAP kinase, then we should find the EGF receptor as an oncogene. There should be human tumors or viruses that contain oncogenes, one of which would be the EGF receptor that's mutant. A mutated EGF receptor should be found as an oncogene. Okay? And then when we express that mutant form of the EGF receptor into cells, it should stimulate the pathway uncontrollably in the absence of EGF and cause uncontrolled growth. And in fact, the EGF receptor was first identified, the receptor for EGF was first identified by cancer geneticists, one of which is Mike Heyman in the microbiology department here, discovered the EGF receptor as a mutant form in a virus. And that mutant form of the receptor was a receptor that was activated even in the absence of EGF. And that led to the understanding of the mechanism of EGF receptor activation. And the way that EGF receptor is activated, well, first of all, what is the structure of the EGF receptor? It is a new type of receptor, very different. It is not a G protein linked receptor. It is a different type of receptor. It doesn't have 7 transmembrane domains. It has a single transmembrane domain. It has an extracellular domain that binds EGF. And it has, again, a single transmembrane domain and an intracellular domain that contains enzymatic activity. The binding of EGF to this receptor stabilizes a dimer, which is active. So the active form of this receptor is a dimerized form of the receptor, which is stabilized by EGF binding. And what happens is a conformational change in the cytoplasmic domain, such that this intracellular domain is a protein kinase that becomes activated in the dimer. What Stan Cohen did was to take hold of this receptor in cells and try to figure out what the substrate was for this receptor activated kinase. And what he found, after painstaking work by many labs, he identified that the receptor is phosphorylating itself. The first substrate and main substrate of this receptor is an autophosphorylation within the dimer. So this encodes catalytic domain of a tyrosine kinase that in the dimer, one kinase phosphorylates the other member of the dimer. And you get multiple phosphorylations on tyrosines within the receptor itself. Those phosphorylations play a key role in the way that this receptor then mediates the activation of RAS and mediates the activation of other effector pathways. Each autophosphorylation site in the receptor is a site of phosphotyrosine to which specific effector proteins can dock, can bind to. And one effector protein that binds to a particular phosphotyrosine is a complex that includes the guanyl nucleotide exchange factor for RAS. And the guanyl nucleotide exchange factor for RAS is brought to the membrane by binding to the phosphotyrosine and brought to RAS so that they can interact and we could activate RAS. So let's look at the steps involved. The tyrosine kinase contains an autophosphorylation site to which an adapter protein called GRB2 binds to by its SH2 domain. So this adapter protein has an SH2 domain that binds to the phosphotyrosine, and it also has an SH3 domain that can bind to the guanyl nucleotide exchange factor for RAS. When these are brought to the membrane by virtue of the existence of this phosphorylation site, RAS can now be activated. So the mechanism of activation is autophosphorylation creating magnetic docking sites for effectors, one of which is this complex of adapter and GEF for RAS. So we now have a pathway from receptor to multiple effectors for growth control, in which, in the case of EGF, but this is expanded to other growth factor receptors that mediate mitogenesis. The ligand binds to the receptor dimerizes, the tyrosine kinase is activated and autophosphorylates. Adapter protein complexed with GEF can now come to the membrane, binding to the phosphotyrosine and activating RAS. RAS would then activate the RAF kinase, which then phosphorylates and activates MEK, which phosphorylates and activates ERK. Now, what we'll learn in the last third of the course is that all of these pathways eventually lead to the nucleus, to change gene expression. And so one of the downstream substrates of ERK is a transcription factor that affects gene expression. And multiple transcription factors are affected, and the mechanism by which gene expression is changed we'll learn in the last third of the course. Also, an interesting aspect of these receptors is that they not only control mitogenesis, they control phenotype, they control the anchorage-dependent growth, they control contact inhibition, and they do so by multiple sites on the receptor having phosphotyrosines to which different proteins can interact. And we're going to learn more about that over the next several lectures as well. Okay, good luck on the exam.